Thank you for the opportunity to speak to you today about new synthesis of homotrimeric type 1 collagen, matrix metalloproteinase 3 and TIMP2 in Dupuytren's disease tissue. Dupuytren's disease begins as a proliferative nodule under the skin of the palm of the hand. This can progress to produce a collagen-rich cord, which contains type 1 collagen. Contraction of the cord, the collagen within the cord by myofibroblasts, can cause the digits to bend over, causing disability for patients. In these advanced cases, surgery can be used to resolve the problem. However, there's high recurrence rates of the disease following surgery. So we hypothesized that this recurrence could be due to a continual synthesis of new collagens and other extracellular matrix proteins. So one aspect that we looked at was newly synthesized type 1 collagen, which is the major constituent of the cord. We looked at the messenger RNA, which is derived from two genes, col1A1 and col1A2. And we also looked at new protein synthesis using 14C proline labeling. There's an abundance of proline residues within the collagen triple helix, and therefore it's possible to pick up new synthesis of collagen using this method. Type 1 collagen is a heterotrimer of 2 alpha-1 and 1 alpha-2 chains. It's formed as a proform called procollagen. The N and the C propeptides are then proteolytically removed to form tropocollagen, which spontaneously assembles into fibrils. So the normal ratio of alpha-1 to alpha-2 is 2:1. However, an abnormal type of type 1 collagen called collagen homotrimer contains 3 alpha-1 chains. If this is produced alongside the heterotrimeric form, the ratio of the alpha-1 to alpha-2 chains increases. So we can extract the collagen from the tissue and run on an SDS page gel and carry out densitometry on the bands corresponding to alpha-1 and alpha-2. And the normal ratio of these bands will be 2:1, but if homotrimer is present, the ratio increases. We've also used a proteomics approach using 13C lysine labeling to label all types of newly synthesized proteins. So we used tissue explants, added 13C lysine, and then analysed the tissue, which was powdered and then extracted, or we analysed separately the media. And in both cases, proteins were fragmented by trypsin and analysed by mass spectrometry. Trypsin is particularly compatible with lysine labelling because each tryptic peptide then contains at least one lysine residue. So we looked at newly synthesized proteins containing heavy lysine. We also did a label-free analysis to look at the proteins that were present in the tissues before the start of the labeling period, so that were not newly synthesized during that time. So firstly, looking at type 1 collagen, we looked at the messenger RNA, and that this was increased for col1A1 and col1A2, consistent with previous studies, but the ratio of col1A1 to col1A2 was also increased, which indicates that potentially this could be driving formation of type 1 collagen homotrimer. But we also looked at protein synthesis. So in normal palmar fascia, there was almost no type 1 collagen produced, but in Dupuytren's tissue, many samples were producing new type 1 collagen. And when we quantified the ratio of the alpha-1 to the alpha-2 chains, we found that this was often over 2, and this could correspond to a percentage of homotrimeric collagen, even up to 50% in one sample. Looking at our 13C lysine labeling results derived from mass spectrometry, reassuringly we found new synthesis of col1A1 and col1A2 in Dupuytren's samples. We also found high incorporation of label into peptides from matrix metalloproteinase 3 and TIMP2 in both the nodule and the cord of Dupuytren's tissue, and this was higher than in normal palmar fascia. We found a similar effect for MMP2 and fibronectin, although a post hoc test couldn't specifically distinguish between groups here. Looking at our label-free analysis, we found a group of proteins in normal palmar fascia, particularly myocillin. Myocillin is of interest because it's a Wnt pathway regulator, and Wnt signaling can be disturbed in Dupuytren's disease. In the Dupuytren samples, we found a large number of proteins were more highly abundant, including collagens, other extracellular matrix proteins, and cellular proteins. And how these proteins interact with each other was visualized using STRING, which is a virtual protein-protein interaction network. So to try and make sense of this interaction network, we looked for upstream regulators using a program called IPA, and TGF-beta came out as one of the top potential upstream regulators of the different protein abundance in Dupuytren's disease. So in summary, we found active synthesis of normal type 1 collagen, heterotrimer, but also homotrimer, particularly in some samples, and of increased new production of matrix metalloproteinase 3 and 2, and of tissue inhibitor of metalloproteinase 2 in Dupuytren's disease. Hence, this new synthesis could be contributing to recurrence and could potentially be targeted to slow or prevent recurrence. We also identified alterations in TGF-beta regulated proteins and a different expression of myocilin, which is a Wnt pathway regulator, and alterations to these pathways are consistent with previous studies and support modulation for potential therapeutic benefit. So I'd like to thank all the staff and students who've contributed to this work, as well as my collaborators, and then the NHS trusts involved, including their surgeons and research nurses, and especially the patient donors who agreed to gift their tissue for this study. Thank you very much for listening.